Hello, everyone. My name is Thomas Desvignes. I'm a research associate at the University of Oregon, where I work with John Postlethwait. And today I'm going to present you some new results about the molecular basis of hemoglobin multiplicity reduction in Antarctic notothenioids. So hemoglobin multiplicity is the expression of multiple hemoglobin isoforms that have different physiochemical properties. And this is achieved by expressing different alpha-globin genes and different beta-globin genes that together combine into different hemoglobin isoforms. And this is important because it helps animals adapt to changing environmental and physiological conditions. One very interesting thing is that Antarctic notothenioids have reduced hemoglobin multiplicity compared to temperate notothenioids. And plunderfish and dragonfishes, which are the most closely related groups to icefishes, they express only a single hemoglobin. And that has been proposed to result from relaxed selective pressure in the thermally stable and highly oxygenated Antarctic waters. So here we wanted to test the hypothesis that changes in hemoglobin gene content or hemoglobin gene expression underlie the reduction in hemoglobin multiplicity in Antarctic notothenioids. So in an article that actually just got published last week, we looked at the gene content in hemoglobins in different notothenioids. And we found that the repertoire was actually well conserved and that even species with only expression of one hemoglobin had numerous alpha and beta globin genes. So from there, we conclude that hemoglobin multiplicity reduction was not caused by hemoglobin gene losses. So we looked at the expression of those genes in a variety of species and in head, kidney, and spleen, which are organs related to the formation of blood. And in our outgroup to notothenioids, the perch, we found that 2 alpha-globin genes were expressed in red here, and 2 beta genes were expressed here, leading to the prediction that potentially 4 different hemoglobin protein could be produced. When we look at the basal thornfish, Cotoperca trigloides, we see that 2 alpha-globin genes are expressed and only 1 beta gene, which is consistent with the non-multiplicity of hemoglobin here in that species of 2 hemoglobin. Now, when we look at Elogenous macrovinus, which is the closest species related, closest to Antarctic notothenioids, we see that there's a dramatic reduction in expression of the MN cluster genes, and at the same time, an increase in expression in the beta-globin gene of the LA cluster. In Antarctic species, like in the toothfish and Trematodus pyrenaicae, we see that there's a further reduction in the expression of the MN cluster genes, which is consistent with their multiplicity that is dominated by one isoform. And in species that are known to have only one hemoglobin, we see that the genes of the MN cluster are not expressed at all, the same that we see in icefishes that don't express any hemoglobin genes. So from that, we conclude that hemoglobin multiplicity reduction is linked to the lack of expression of MN cluster genes. But we also looked at the expression of hemoglobin genes in embryos. And here in the perch, in adults, we predicted 4 hemoglobins. And in the adult dragonfish, only 1 hemoglobin. In embryos of perch, we predict 6 hemoglobin, the combinations of 2 alpha and 3 beta globin genes. And in the dragonfish embryos, we predict the same, 6 hemoglobin, there by combinations of 3 alpha genes and 2 beta genes. But that leads us to propose that, or to predict that embryo of dragonfish have a much higher hemoglobin multiplicity compared to adults. So in conclusion, we showed that hemoglobin multiplicity reduction was not caused by gene loss in Antarctic notothenioids. Instead, it's the beta gene from the LA cluster that becomes predominantly expressed, and the genes from the MN cluster that are lowly expressed or not expressed at all anymore. The finding that dragonfish embryos express multiple hemoglobin genes suggests a high hemoglobin multiplicity and led us to question whether sensitive embryos and larvae may survive best viable conditions when they express multiple hemoglobins, while robust adults maybe don't require many hemoglobins to handle variations to the environment. So thank you very much.